Hi everyone!
You're listening to SciPub,
where we are talking science.
Not only for the experts, but for everyone.
Welcome to a new episode. Hello everyone, this is Gorane.
This is Mo.
And you're listening to SciPub. Today we are more than happy to present our guest, Natalia Roson, a PhD student in IS Global. And our topic today is microbiology. So thank you for coming and having your time. So let's start.
Thank you for having me.
So first, could you explain in simple words what microorganisms are?
Yes, so microorganisms essentially are very tiny, small living beings that cannot be seen for human eye, and you can only see them under a microscope. This includes bacteria, as you may know, some algae, some fungi, some protozoa, which are like primate eukaryotes. This includes also parasites, like the malaria parasite, and also viruses, although the scientific community really don't consider viruses as a living being, but it is a microorganism.
Okay.
Yes.
So, not all microorganisms are bad, right? So, can you explain this a little bit?
Yes, so not all microorganisms are infectious causing agents, or infection causing agents, if that's what you mean. There are microorganisms everywhere. Everywhere you can think of, there's a microorganism living there, like from your skin to volcanic areas to deep under the water, like 300 meters in the deep ocean. You can find microorganisms. Most of them are environmental microorganisms, and they have very useful functions in nature, like helping growing trees, like fungi, bacteria, also keeping in balance the community in the soil, and also it's the same in humans. We have what we call good bacteria, which is our gut microbiota, and they help us stop opportunistic pathogens that may cause us a disease.
So, antibiotics are used to fight against these bad bacteria, right? But how do they work?
Yeah, so antibiotics are therapeutics that we use to treat bacterial infections. The important part about antibiotics is that their target, what makes an antibiotic a good antibiotic, is that its target, it's something specific from a bacteria. It can be the cell wall, it can be a lipopolysaccharide, it's a very difficult word, that it's expressed in the surface of the bacteria, and this is very important because otherwise it would be also toxic for us. So the important thing is that they tackle bacterial elements and not human elements. Basically, antibiotics are produced by organisms, by living organisms. We have also, because science has evolved a lot, and we are able to synthesize artificially antibiotics, but in its beginning, when it was discovered, antibiotics are produced mainly by bacteria and fungi to fight for a niche in the environment.
Okay, yeah. To fight against
each other, like for the place to live.
And what are pre- and probiotics?
So we have two different things here. Firstly, we have the prebiotics, which are molecules. Specifically, they used to be like carbohydrates or other nutrients that we eat, like fiber, to help the bacteria in our gut to develop and to create what or we want it to be like a healthy environment in terms of community and like a very diverse microbiota. And probiotics in contrast, are living beings. They are bacteria and yeasts, like Saccharomyces or others. And the base or the idea in probiotics is that they help to recover and to promote the diversity in our microbiota. However, I have to say that there is not much evidence that they really do that.
Interesting, thank you. So what happens, like it's important to use antibiotics correctly. So what happens if people take them in an incorrect way?
It happens a lot of things. So I'm glad you make this question because there's not much knowledge in the general public about what antibiotics are and how to take it. Like for sure, if you look at the medicines you have at home, for sure there will be some antibiotic in there, and it will be expired. 90%, I'm sure it will be expired. So I'm gonna put you an example. Okay, like for example, when you have an infection and you start taking antibiotics, firstly it has to be prescribed. We cannot take antibiotics because we want or because we have it at home. Our doctor needs to tell us, like we need the doctor to tell us that we have to take these antibiotics, because sometimes we have a flu or we have a cold, and which is caused by viruses, and people take antibiotics because they think it will work, but it won't, because antibiotics only affects bacteria, and for viruses we take antivirals.
Let's make that clear.
Yes.
And sometimes for these common infections or viral infections, we don't take anything because they just happen and then you recover. So when you have a bacterial infection, it's very important that you do a good use of that antibiotic. That means that you take the correct amount of antibiotic during the time that the doctor tells you to take it. Like if you have to take a 7-day, 3 times a day, you have to do this and you cannot stop. Like it's very important because the fact that you don't have symptoms doesn't mean that you are completely healed. Like maybe you need this bacterial load to develop symptoms, and then you start taking antibiotics and the bacterial load comes here, but it doesn't mean that there's no bacterial load. If you stop taking antibiotics before this bacterial load comes to 0, what happens? That this will grow again and we will develop again the infection. And what happens in this scenario is that some of this bacteria has been previously exposed to this antibiotic, and when we take what we call sub-inhibitory concentrations of this antibiotic, that's what happens when we don't take all the treatment, they are more prone to develop mutations that makes them resistant to this antibiotic. So each time we don't take in the correct way the treatment, it's more easy. The easier is that this infection is resistant, and that we need to take another or a different, to go again to the doctor, have another prescription, and take another antibiotic.
Okay.
And I would like to take from this point is this is related to antibiotic resistant. Is how it's made. So can you explain what's antibiotic resistant or antimicrobial resistant?
Yeah, so it's very important to know that antibiotic resistance is a natural, it's a natural process, and it already exists in nature, because as I said, they use antibiotics to fight each other, and then the other one develops the resistance, and then they are continuously evolving. And what happens when we overuse antibiotics is that we are accelerating the process of bacteria becoming resistant. The issue now is important because since the discovery of antimicrobials, we have used them. We were not that aware of the issue, and we used antimicrobials for everything, to treat animals, to give to animals as growth promoters. But now it's forbidden, at least here in Europe, and this led to the development in a very heavy way of resistance. But it's natural, and it happens when a mutation occurs that makes, I don't know, there are a lot of ways of bacteria becoming resistant. There are a lot of mechanisms. But essentially are mutations or genes that are passing from one bacterial to another.
Yeah.
So how much it's hard scientists trying to find alternative for this resistance, because if you have a bacterial resistance for antibiotic, maybe there's some cases it's end with this. So how much the situation is hard? Are we in very bad situation, intermediate, or we are still at the beginning?
It's hard because it's very hard to develop new antimicrobials. So bacteria are very clever and they become resistant very easily. And now, since the last decade, we don't have any new antimicrobials. This means that we don't have new modes of action, new targets. We do have new versions of all antimicrobials, but what happens if the bacteria has been previously trained to this type of antibiotic, it will become resistant. It will work at the beginning, but the resistance will come very, very faster comparing to a completely new antibiotic. And it's very hard also because funding, it's an issue. And it's understandable that big companies do not make profit from researching and developing new antimicrobials. In the end, they are companies, and the main goal is to get profit. At some point, it's governments and public funding who takes this big responsibility to develop, mainly in the first stages of the research, to develop and to find and screen molecules and see if some of them has antimicrobial activity, and after that you go to in vivo models in mice or rats or whatever. In case it's not toxic to animals, it has very good pharmacokinetics, pharmacodynamics, and you can go to clinical trials. There's a very, very, very huge bottleneck that makes almost all of them fall in the first phase or second phase. And not saying the third phase. So it's very, very difficult that it finally comes out to the market. And also to have this molecule in clinical trials, you also need funding and volunteers, and that's very difficult to have too.
So it's very complex process to generate a new. And for that we should take care when we take antibiotics, because it's like we are not so close to find and to solve the problem. See.
We have to slow down a little bit this use, so we can have them the longest time, like the more time possible.
This is an important point. So now we would like to know, what are you doing now in your PhD? Which kind of research you are doing?
Okay, so what's my PhD about? So I work, as you already introduced me, in the Barcelona Institute for Global Health. And I belong to the Antimicrobial Resistance Initiative. So we are a research group that makes different things. In my case, I kind of do drug discovery. I just try different molecules and see if check as I told you before, like the first stages of the research, I do antimicrobial activity testing. I just like trying to figure out which is the target or the mode of action, how well it interacts, or if it affects like I don't know, human serum. Like these are kind of things that should be taken into account when you are trying to develop a new molecule. And we are on it now. We also have collaborators that do the in vivo models for us. Like in science,
Like between computational and experiment, or it's not including computational work.
We really don't do computational work. Like, but there are very interesting strategies now coming up with artificial intelligence and machine learning. Yeah, like training the machines to become like to predict if among a pool of molecules which one could have activity or not, or if it could be toxic or not. Yeah, that's a thing. It's very interesting, and it definitely will make the work very easier. With the bacteria that I'm working, the type of bacteria that we manage, we only work with bacteria, starting from them, because antimicrobial resistance and to antibiotics involves bacteria. And specifically, I work with some pathogens that are considered by the WHO, the World Health Organization, as criticals, or as the ones that we have to focus on and invest our resources to find new antimicrobials. These are, and the whole group mainly focus on this, what we call SciPub. It's an acronym of Escherichia coli, Staphylococcus aureus, you may know, Pseudomonas aeruginosa, Cinetobacter baumannii, Bacter, and I think I have Klebsiella pneumoniae. Specifically, they are very critical when they become resistant to carbapenems, which are the last resort antibiotics we use, like the last option we have to treat bacterial infections. And when we have isolate in the hospital bacteria that are resistant also to carbapenems, this is very worrying. Because and sometimes this makes you recover antibiotics that initially we stopped using them because they were toxic, or that's the last options we have.
I have a question, because I remember when I was studying, I'm not expert in microbiology, but you said Escherichia coli is classified one of the critical ones. You should take the resources. I think it was good one, or it's worse for us.
It depends on the strain. The one that is critical is the Escherichia coli resistant to carbapenems. Yes, so, and the ones that can cause infection, but you can also find Escherichia coli as probiotic, or we use, in fact, it's a very useful bacteria for research and for genetic engineering. That's what we use when we want to study, I don't know, a gene or express something, I don't know. It's very, very easy to manage and to grow, and it's like the most famous bacteria. But there are also pathogenic strains.
So you mean not just, you can say all of the Escherichia coli is good, or all of them they are bad? It depends on the strain?
Never is black or white. There are grays in the middle. And among the pathogenic Escherichia coli, there are some of them that has a toxin that makes the symptoms worse, and others is just like causing diarrhea, but you don't use it. It's like it depends.
So you may know that we are going to the streets to ask people with their doubts or questions. Our concern started when we were asking if they knew about antimicrobial resistance, and every answer was a no. So, but they had different questions. So one of them would be, well, you already answered, but what kind of infections can be treated with antibiotics? Like everything.
I think you can never say this too many times. Like antibiotics treat bacterial infections, and in fact, I'm not surprised that people out there doesn't know or don't know about this, because even me, before starting working on this, and I'm a microbiologist, I knew about antimicrobial resistance, but I didn't know it was that important or that worrying. So if that's happening to me, that I must study something focusing only in that, maybe someone up there that it's completely out of this world of microbiology. But there's where scientific dissemination enters, and I think it's very, very important. And it's very, very, very important what you do here. I hope this gets a lot of views, because it is an issue, and people should know about this. And I'm talking to my family and my friends all the time. The other day I went to a friend's house, and I saw that she had like different antibiotics on in the kitchen. I was like, why do you have this here? And she told me, if I feel bad, I just take it here. Oh no. Okay, I will give you a lesson. And I just told her, I will take this. I will take this out of your house. Is that okay? Yeah.
No, yeah, it's true that we should make everyone know about it, because if everyone does a little bit, we could.
I think the important thing here is to know that that antibiotics must be prescribed, and once you finish taking all the treatment, if there are some pills or some rests in the box of antibiotics, you have to take them to the pharmacy. Don't keep them at home, let them expire. If you have in 1 or 2 years another infection, the doctor will give you another prescription, because you need it. So don't keep them at home, just throw them in the pharmacy, because throwing in the regular I don't know how it says, the trash. And you are helping them to arrive to the environment, and we don't want that either. To have environmental strains also resistant to antibiotics.
Okay, so for me, this is one of the best parts in the podcast, because it's not just for public, but for us, and also for young researchers, for the next generation of researchers. So could you tell us about your career, how you started to love science, decided to take a science as a career, and so on?
How I started loving science. I like everything. Actually, I really struggled when I had to decide what to study, this decision that you have to make when you are 17 or 16 years old, and you have to decide between science and letters. And I was sure I wanted to do some kind of science, but I didn't know. I liked also biomedical engineering. I liked architecture. I was into everything, but biology is something that I really like. And at the beginning, I wanted to study medicine, and now I'm not a physician, because I didn't have the grade. I have to be honest. But I knew I wanted to do something related to the clinical world, so I decided to start with microbiology, and I loved the degree in the university. I came from Bilbao to Barcelona to study here the 4 years, and then stayed for a master's. I did also in the IS Global in collaboration with the University of Barcelona, and there is when I met the group I am working with now. I started with my final master's project, just the same way you are doing now. I really I entered the group very well. I liked the work I was doing, and my supervisor offered me the opportunity. More than an opportunity, they offered me to start asking for grants and to search funding, because in science it's very difficult to find funding for everything. So I started 1 year when I presented the master thesis. I spent 1 year asking for grants. Luckily, in that year, I had a grant from the Basque government, and I went to Scotland 6 months to work there in a laboratory. It was in an environmental laboratory, completely different from what I was doing, but because my master's was kind of related to global health and international health, I really enjoyed that too, because we were finding resistance genes in the environment, so it was very interesting too. In June, the pandemic happened, and we had to stay in Scotland, not working, not doing anything. Yes.
But it was related to public health too, so it was a time to study this kind of topic, so it was amazing, huh?
Yeah, totally. But in June I received a grant from the UB, the University of Barcelona. And now I'm on my 4th year of PhD, about to finish. Yes, it's difficult coming to here. I can see the light now. At the beginning it was a bit hard, but now.
Since you are close to finish your PhD, so what are you planning for your future? And I know this kind of question.
Yeah, it's very difficult. Sometimes you don't know. Sometimes the opportunities comes, and you have to decide if you want to take it or not. For sure, you need to have a focus on something. In my case, because I told you it's very difficult to find funding in the academic world. I'm not sure if I want to spend the rest of my life asking for grants, new projects. I don't know. Now it's I'm more focusing on industry, but because I think it's the way you can things come into life, from the laboratory to the real world. And that's something I like. Maybe it's very utopic, because I know in industry I won't be able to develop.
Sorry, but more money also. So it's
well, that's a thing. That's a thing too, and more. I won't say money, but stability.
Yeah. Okay.
Yeah. But even that, you never know, because you can lose all the jobs. So sometimes you don't know. But it's a thing that here you don't know what you will be doing next year if you don't find a new project or a new something that gives you money to keep you on your team, because that's a thing too.
More or less, it's a final question, but would you like to give an advice for young or new generation, young researchers, or also for public?
This is very difficult too. But I think it will sound very, I don't know, but you really have to do what you are into. You won't enjoy working on something you really don't like. So the first thing is that if you like research, if you like science and contributing to the scientific community, this is definitely your path. And the fact that it's not easy, it shouldn't stop you from doing it.
Yeah.
You know, because there's a lot of people working on that here and they made it through. So yeah, if you like it, just go for it. The thing is that in my case, I don't know if I want to stay, but like I have a lot of friends that love researching in the academic world and wants to stay, and they have like things very clear. And also, I don't know, if you like microbiology, then do microbiology. If you like engineering, then do engineering. There's a lot of, there's a very wide range of options.
Yeah.
And if you start something and you realize that you don't like it, what stops you from starting change and starting another?
This point is more or less a lot of our say they are saying it's like, don't be shy to change your not career, but you still have a time to do it. Why not? It's important.
I think in a professional career, the line is not like that. You will have failures, and you will receive a lot of no's, and you will go like this, and then you go again, and you see. I received a lot of no's before I had the final grant that allowed me to start the PhD.
And any final word like to end the interview, like don't take antibiotics, but yes.
Don't take antibiotics if your doctor doesn't tell you to. So yeah, I don't know, I had fun. I hope you do also.
No, we enjoyed a lot.
Yes, thank you. It was very interesting.
It's a very interesting topic. I'm very happy to have you.
Yeah, it was very nice. Thank you.